everyone. Hello everyone, I'm Pooja Jaiswal and I'll be presenting my topic of evaluating the reproducibility of bioinformatic tools for adaptive immune receptor repertoire analysis. So, what is TCR receptor? So, TCR receptor are made up of two heterodimers and which consist of an alpha chain and a beta chain, or it could be a gamma or delta chain. The alpha beta chain comprises of 95% of the TCR cell, and the gamma and delta chain is only 5%. So, this chain are made up of three or four different type of segments, which is V region, J region, C region, D region. The D region is present only in the beta chain. So, what makes this TCR receptor unique? For each type of antigen presenting cell, this TCR receptor undergoes a unique recombination and it produces a different kind of TCR cell for that particular antigen presenting cell. So, it's like based on this recombination, which is 2 into 10 raised to 19 unique TCR receptors which is found, this TCR receptor is very diverse and it is responsible for the adaptive immunity of us. So it is very important to study this AIR TCR receptor sequencing reproducibility. Bringing that to the topic of concern, like why reproducibility? Since the data is highly diverse and this VDJ recombination produces this vast amount of unique clones, it is very important to select the tool which can give us consistent and reliable results. So for that, the selection of tool, it totally depends on the tool's lifespan, the current version of the tool which you are using, the parameter setting of the tool. So if keeping all of this constant and if you are doing multiple analysis of same particular sample, you should get exactly similar results. That's the end goal. But do we get that? That's the main question. So how do we know that the tool which we have selected is working correctly or not? It highly depends on the accuracy and efficiency of the tool. If the tool is able to correctly identify the reads and align it to the VDJ genes and generate the clonotype for all these samples correctly and same, then it means that our tool is working perfectly and it is producing reproducible results and it is hence reliable. Coming to how we are going to study this reproducibility is that whenever we do any kind of sequencing, we prepare the library, be it DNA sample or RNA sample. This library preparation step also introduces some error, human error or the machine error. And this will generate technical replicates. After sequencing, we'll get the technical replicates, and this technical replicates will be generated by multiple people by running in multiple labs, and this ensures the method reproducibility. So technical replicates are generated by sequencing same sample multiple times, thereby showing that the results which we are getting is reproducible. The another method to perform this analysis is by introducing synthetic replicates, which the replicates which we are generating in silico by computational power. Even though it does not replaces the technical replicates importance, but it can help us in understanding whether the bioinformatic tool in question is working correctly or not if we keep the parameters same and not. This synthetic replicates can be generated just by shuffling the reads or preparing its reverse complement, and then we check whether the tool is reproducible or not. Coming to the preliminary analysis, for example, if I ran my data on one particular tool and then I found out that the alignment rate technically for original replicates of 51.47, I should have gotten similar result for the resorted rate. If I'm not getting it, means there is some kind of error which is introduced through a manual error or the tool error. So we need to identify what kind of error it is, and it will help us in generating reproducible results which will make it more reliable. As you can see, the VDJ region coverage for original rate was 71.5, but for the resorted rate, it drastically dropped. And the clonotype which was generated, it was 254.31, like 25,431, but now it's only 172. It means there is some error with the tool, and it makes us question whether this tool is reliable or not. So the UMI count has drastically reduced, which is clearly visible in the top 10 clonotypes, which is generated by both original sample and resorted sample. Technically, it should be very similar, but it's not in this case. So it means like we have to identify why synthetic replicates are important in reproducibility. So technical replicate, it captures the diversity of the entire repertoire, but it is very prone to error and through multiple factors, like it could be human error, error in library preparation. But synthetic replicates are fast and cost efficient way of generating replicates. It will not replace the technical replicate, but it will help us in understand whether the tool which we are using for bioinformatic analysis is reliable, reproducible, and efficient. And this will help researchers in identifying what's the most reliable tool they can use in terms of reproducible result. And so this study focuses on addressing this common sources of variation and help the researchers. So thank you.